Dear students, today I am going to tell you about the, well, first of all, I want to tell you that this is for our course Cell and Molecular Biology. Course code is BBBMBT1002, and today's I am going to discuss about the mitosis. In the last class, I introduced you with cell division and cell cycle, and we know there are two types of cell divisions, mitosis and meiosis. Now, today I will be discussing about the mitosis. Okay. Now, this cell, this slide tells us about the mitosis. It has got two divisions, division of the nucleus, which is called the karyokinesis, and then that is followed by the division of the cytoplasm is known and it is called cytokinesis. Now, this slide is showing the division of the nucleus or karyokinesis, and this occurs in eukaryotes. It has got these four stages. It doesn't occur in cells such as the brain cells. So this stage is known as prophase. When under the microscope the chromosomes becomes clearly visible, and this is then followed by this stage known as metaphase, where all the chromosomes arrange in the equatorial region of the cell, and then this stage comes, the anaphase, when the chromatids separate and move to the two poles of the cell, and here it divides into, nucleus divides into two. And this is followed by the cytokinesis. This slide, it has got four mitotic stages: prophase, this is prophase, this is metaphase when they arrange in the equatorial region, this is anaphase when they start moving towards the pole, and this is the telophase when the nucleus or the chromosomes have divided equally into two, and these are the two nucleus formed, and later on the division of the cytoplasm will take place. Now, this is the microscopic. How do early prophase look under the microscope? Chromatin in nucleus condenses to form visible chromosomes. See, this is cytoplasm, the nuclear membrane. This also becomes slightly invisible. This disintegrates, and this is nucleolus, and these are the chromosomes. And now mitotic spindle forms from fibers in the cytoskeleton or centriole in the animals. Now, this microscopic slide shows the structure of the structure of the nucleus in late prophase stages. So, nuclear membrane and nucleolus are broken down. The nuclear membrane which surrounds the nucleus disintegrates, and nucleolus also breaks down. Chromosomes continue condensing and are clearly visible. Spindle fibers called kinetochores attach to the centromere of each chromosome, and spindle finishes forming between the poles of the cell. Now, this is the diagrammatic presentation of how does it look, how the chromosome attaches to the kinetochore fiber or spindle fiber. This is the kinetochore fiber. Kinetochore fiber are those astral fibers which attach to the chromosome, and they attach in the centromeric region, or which is to the center of the chromosomes. And these are the two chromatids of the chromosome. Now, this figure shows the review of prophase. This cell is showing the prophase stage. The cell looks like this during prophase. Everything remains the same. The chromatids become very clear. The chromosome become very clear in the prophase. And this is the next stage. Mitotic spindle begin to form. So the centriole divides into two. These are the two centrioles, and this is the spindle fibers. And now this is the next stage when the centromere and kinetochore, they look like this. The astral fibers to which the centromere is attached are known as kinetochores. So these are the kinetochores to which the two chromatids of the chromosomes are attached. Now, nuclear membrane. So what are the highlights of this? Nuclear membrane disintegrates and nucleolus disappears in this stage. Now, chromosomes condense, mitotic spindle begins to form and is complete at the end of the prophase. Kinetochores begin to mature and attach to these are the kinetochores. They attach to the spindle. These are the spindle fibers. Now, this is the diagrammatic presentation of the spindle fibers. The mitotic spindle form from the microtubules in the case of plants and centrioles in animal cells. The centriole divides into two. So these are the two centrioles from which arises these fibers or astral fibers. Polar fibers extend from one pole of the cell to the opposite. These are the polar fibers. They don't attach. Polar fibers, those astral fibers which are not attaching to the chromatids or chromosome are known as polar microtubules, and kinetochore fibers are those astral fibers which attach to the chromatids. So this is the kinetochore. Kinetochore fibers extends from the pole of the centromere of the chromosome to which they attach. From these two poles they are attaching. So this is kinetochore. And asters are short fibers radiating from the centrioles. These are short fibers. They are not end to end. So they arise from the centrioles. Now, this slide is showing this is the slide. How do they look under the microscope during the metaphase? We know that during metaphase, the chromatids arrange in the equatorial region of the cell, of the nucleus. So this is now chromosomes attach to the kinetochores. These are the kinetochores to which the chromosomes have attached. They move to the center of the cell, and chromosomes are now lined up at the equator. So these are the chromosomes. They have attached during the metaphase. What happens? They have the already attached chromosomes. They attached with the kinetochores. These chromosomes arrange themselves in the equatorial region of the nucleus, and this is how they look under the microscope. Let us come to our. This is Anaphase. This slide is showing the Anaphase. This occurs rapidly. Sister chromatids are pulled apart to opposite poles of the cells by kinetochore fibers. So what happens during the anaphase? The kinetochore fibers start contracting and they start moving towards the two poles, two centrioles, and they move towards the two poles. And it is all because of the contraction of the kinetochore fibers towards the pole, and they move. So the chromatids are drawn towards the two poles. So this is the diagrammatic presentation, how they are drawn towards the poles, the chromosomes. Now, this figure shows the anaphase review. Now, what happens during the anaphase? Centromere divides into two. Spindle fibers pull sister chromatids. These are the spindle fibers, and they are pulling the sister chromatids towards the two poles. So spindle fibers pull sister chromatids to opposite poles of the cell. Now, each pole is basically, it becomes the future daughter cell, and number of chromosomes are identical in the two cells, or the genetic makeup is exactly same in the two cells. Now comes telophase. Sister chromatids are at opposite poles. Spindle disassembles in this stage, and nuclear envelope again starts forming. So actually the nucleus, now it is divided into the two nuclei, or two nucleus. So spindle disassembles. Now nuclear envelope or nuclear membrane also start forming around each of the sister chromatids, and nucleolus, which disappeared during the early part of mitosis, also start forming. And then this is followed by cytokinesis. So all these stages, prophase, metaphase, anaphase, telophase, all they come under mitosis or karyokinesis. Karyo means nucleus, and karyokinesis has all these four stages, and it results into division of the nucleus into two, and this stage, karyokinesis, is followed by the cytokinesis, or cytokinesis is basically the division of the cytoplasm. Now, so during telophase, telophase is followed by cytokinesis. Chromosomes reappear as the chromatin, as they were present earlier. Now, here we are comparing the anaphase and telophase. This is anaphase. This is basically how do they look under the microscope. The anaphase stage, and this is telophase. So during anaphase, as we know, the chromatids which during metaphase, which were arranged during the equatorial region, start moving towards the two poles, and this is anaphase. And during telophase, what happens? They completely come to the two poles, and nucleus is completely divided into two, but having the same genetic makeup. Now, and this is the diagrammatic presentation of this anaphase, and this is telophase. So what are the highlights of the anaphase? Kinetochore microtubules shorten, separating chromosomes to opposite poles. So what happens? The kinetochore microtubules, they start shortening and start moving towards the two poles, and they separate chromosomes to opposite poles. And polar microtubules elongate. These are the polar microtubules which are continued from this centriole to this centriole. They also start elongating, leading to the separation into two. And they start preparing for the cytokinesis, because this event takes place for the cytokinesis. Ultimately, cytoplasm will also have to divide. So, and this the division of cytoplasm, as I have told repeatedly, is known as cytokinesis. So this is the preparation of cytokinesis. And during telophase, what happens? The nucleus, the chromatids have moved to two ends and two poles, and now the nuclei starts reforming. The polar microtubules, they start separating and start elongating and separating the whole nuclear setup into two. So this is the diagrammatic presentation of the same. Chromosomes reach poles of the cell. Kinetochores disappears. The kinetochores, the microtubules which were carrying the chromatids to the two ends have disappeared. Polar microtubules continue to elongate, preparing the cell for the cytokinesis. So these are the polar microtubules. They start elongating and ultimately separating into two. And this is for the preparing for the cytokinesis. And now the nuclear membrane reforms. Nuclear membrane starts, which actually dissolved during the early prophase, now again starts forming. Nucleolus, which during the prophase disappeared, now starts appearing in the two sides, in the two sister cells. And chromosomes start decondensing, again start coming to the earlier position, what was the position in before prophase. So cytokinesis means division of the cytoplasm. It is the division of the cells into two identical halves called daughter cells. In plant cell, cell plate is formed at the equator to divide the cell, and in animal cells, cleavage furrow forms in the center of the cell. Now, this slide is showing the cytokinesis. This slide is showing the cell division in plants. This is cytokinesis in plant. Here, in the case of plant, the cell plate is formed, separating the whole cell into two. And in the case of animal cell, the cleavage furrow start forming, and the leading to the division of the cytoplasm into two. So, two identical daughter cells having the same genetic makeup are formed. So that was all about the mitosis. So we have seen that a mother cell divides into two same type of cell having the same genetics makeup, and how this division takes place, that we have studied. It is divided into prophase, metaphase, telophase, and ultimately anaphase and telophase, and ultimately cytokinesis, which is the division of the nucleus. So we can divide the whole division mitosis into two: karyokinesis, which is the division of the nucleus, and the cytokinesis, which is the division of the cytoplasm, or ultimately the complete division of the mother cell into complete two same type of daughter cells having the same genetic makeup. But So this happens in the normal mitosis. But what happens when the mitosis is uncontrolled? If mitosis is not controlled or unlimited cell division occurs, it it causes into dangerous tumors. And oncogenes are special proteins that increase the chance that a normal cell develops into a tumor cell. So these are the references we have to study. So this was all about the mitotic cell division, where a mother cell divides into the two daughter cell, identical daughter cells, which are having the same genetic makeup, or the mother cell having the 2n number of chromosomes will divide into two daughter cells having the same 2n number. So this was all about mitosis. In the next class, I'll be discussing about the meiosis, and you go through these following references.